Good morning. Please welcome honorable speaker, distinguished guests, ladies and gentlemen, to our second plenary session. This morning, we will continue International Conference of Life Science and Biotechnology 2017. In this session, there will be 3 invited speakers. They are Professor Shinichi Nakano, Professor Inocencio Ibuot, and Professor Bambang Sugiharto. The first session is plenary session. The first speaker is Professor Shinichi Nakano from Limnology, Kyoto University, Japan. And the moderator for this session is Mr. Ahmad Saifullah from Chemistry Department, Faculty of Mathematics and Science, Universitas Jember. Mr. Ahmad Saifullah, time is yours.
Ladies,
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi Bapak Ibu, saudara-saudara para peserta conference. Good morning ladies and gentlemen, all the participants for the second ICOLIB International Conference on Life Science and Biotechnology. In this day 2 and the second day, hope after you have a good rest last night in Jember, we will have this morning session we will have 2 presenters. One is Professor Shinichi Nakano and Professor Inocencio Ibuot. Actually, for each presenter, we will have 45 minutes given by the committee. And Professor Nakano actually been scheduled to give this morning a session, maybe in your old timetable, he should present his paper at 11, I think, but because he has to leave for Surabaya this afternoon, so he given this morning session. Okay, ladies and gentlemen, the Professor Nakano will present paper long-term change in water quality in Lake Biwa with special reference to organic matter dynamic, microbial, and ecology diversity. Okay, Prof. Shinichi Nakano from Center of Ecological Research, Kyoto University, Japan. Okay. Today's presentation, as usually we have, like yesterday, will be followed by a question and answer section. And I hope it will take about 10 minutes. So not to waste more time, so I give to Professor Nakano to present your presentation now. I hope it will be completed by 40 minutes. Thank you.
Terima kasih for your introduction. Okay. Selamat pagi. Terima kasih. Yeah, selamat pagi. I am Nakano from Kyoto University, Japan. So today I'd like to talk about this topic. And yes, let me introduce the outline of my talk today. I have to explain what's going on in the water quality of Lake Biwa first, which is laser pointer here. Okay, this one. And followed by my main topic, my research topic, the organic matter dynamics, microbial ecology, and the diversity in Lake Biwa. Okay, let's go. Firstly, I have to explain you where Lake Biwa is. Lake Biwa is the biggest, largest lake in Japan, most socially important lake in Japan. And this is the Japanese islands here, and the famous Kyoto, old capital here, Osaka here, Hiroshima here, Tokyo here. And Lake Biwa is located within the Shiga prefecture. Shiga is next to the Kyoto prefecture. Okay, this is a satellite photo of Lake Biwa, how it looks like. Okay, and sorry. Okay, let me explain the Lake Biwa, the surface area, and the depths and the volume. The surface area of Lake Biwa is 670 square kilometer, and the maximum depth, the 103 meter in the northern basin. We have two basins for Lake Biwa. One is the main basin, this one from the north here. This is the main basin, north basin. The average depth of the north basin is 44 meters. And this is a shallow basin, southern basin. The average depth, southern basin is 4 meters. And this is the most socially important lake in Japan because the 14 million people use this water for drinking, for the industry, for leisure, or for everything. So without Lake Biwa, Shiga Prefecture, Kyoto Prefecture, Osaka, Hyogo, Nara, those people would die because this is a very important lake. And let me introduce the land use of the Shiga Prefecture like this. You can see here many red dots here. This is the old city, capital city of the Shiga Prefecture, and the red dots means the urbanized area. And you can see here the many red dots here, the shallow small basin, southern basin. This means the eutrophication. Do you know the eutrophication? The many nutrients due to the human activity come into the lakes or maybe some oceans. And those nutrients would have the massive growth of the phytoplankton or algae, and the water gets turbid or greenish and the polluted or something. And the eutrophication in the southern basin easily proceed here, but as you can see here, not so many dots, red dots, and here, northern basin. So a northern basin is big and deep and less populated compared with the southern basin. So the eutrophication in the northern basin is still not so serious in Lake Biwa. And as you can see here, this is the wastewater drainage system development in Japanese average, pink one, and the blue one is Shiga Prefecture average. As we can see here, since the 1970 to 1990 or something, the sewage system in Shiga Prefecture is lower than the Japanese average. This means the wasted water in Shiga Prefecture directly came into the Lake Biwa. That would cause the serious eutrophication in Lake Biwa, as you can imagine. But after the very, very big effort by Shiga Prefecture together with the general public and the Japanese government, the sewage system in Shiga Prefecture got better, much better than the Japanese average. And as we can see, this is a figure of the transparency, sedge depth changes since the 1920 to the 2000 something in Lake Biwa. Do you know the transparency? Aquatic ecologists or aquatic researchers measure their clarity or cleanness of water. Firstly, measure the transparency or search depth. We dip the white circle into the water, and if the white circle can be seen the very deep part, this means the water is very clear. But if the water is very turbid, this white disk would disappear soon. So high transparency means that the water is very clean. The low transparency means the water is turbid or something dirty. Okay? So as we can see here, since the 1920, transparency get worse. And then from the 1960 or something, we had very bad transparency here. And then increase again. Okay? So, okay. Let me introduce some eutrophication of Lake Biwa. In 1959, we had some filtration problem in the Kyoto City water treatment plant. So if we have massive growth of the phytoplankton, we have some clogging of the filtering system in the water treatment plant. So in 1959, we had that. And followed by the 1969, we had a musty odor, very smelling water in Lake Biwa, even if after the water purification. And then 1977, we had a red tide, Eurogreen americana, freshwater red tide, not marine red tide. Freshwater red tide is not so toxic, but smelling bad. And in 1983, we had the microcystis brew. Microcystis is a cyanobacteria, not the eukaryotes, cyanobacteria. Some of the microcystis is very highly toxic, sometimes lethal for the human being. Okay? We had the microcystis bloom in the shallow south basin. As you know that this southern basin is easily eutrophicated, so first we had this bloom in southern basin, and this is not so serious picocyanobacteria, but then finally we had the microcystis bloom even if in the northern basin. As you can see, already seen that the northern basin is a big, deep, and not so highly populated. But even if so, we had a microcystis bloom in the northern basin. And let me show you how the red tide look like. Sorry, doesn't work. I would like to show you a video. Can you? Oh, okay. Moving. Okay. This is the video taken from the helicopter, taken by the NHK, and you can see here the many reddish color on the surface of Lake Biwa. It's a red tide, akashio in Japanese. And take a look closer. When we dip the transparency search depth, and we can see here the reddish one, and very, how can I say, very dirty. And when we observe those ones under the microscope, you can see here many yellow balls. These are Eurogreen americana. And this is a phytoplankton. When we leave this one on the microscope, they get together after 1 hour, like this. And if we get closer, take a look at the microscope, this is the Eurogreen. This is the colony of the small cells. Each colony consists of many cells, maybe 200, 300 more. And each cells are flagellates. They have the flagella to swim. And when they become bloom, they blooming by photosynthesis. But after coming to the maximum, they stop photosynthesis and eat bacteria. So the Eurogreen is something like both the plants and the animal. They can conduct a photosynthesis, and they can conduct the eating, grazing on bacteria. Okay? So, okay. And the next one is cyanobacterium bloom. Maybe some of you have ever seen that this is on the surface, the greenish and dirty, very dirty. This is what cyanobacterium bloom, microcystis bloom, occurred in Lake Biwa last year. And some, not all, some microcyclic bloom is highly toxic. So if you have this type of water, please do not touch. Please do not drink. Of course, I think that you do not drink this one, but please avoid touching or drinking this water. After purification, no problem. After purification, no problem. But yeah, please avoid. So the Shiga Prefecture and also the Japanese national government has been making a big effort to improve the water quality of Lake Biwa. And yeah, first one was conducted in 1969, Pollution Prevention Control Ordinance by Shiga Prefecture government. And at that time, the Japanese government did not know how to solve the water quality problem in Japanese lakes. But Shiga Prefecture has some high-quality researchers, not only in Kyoto University, but also in Shiga Prefecture itself. So the Shiga Prefecture could consult with those good researchers. And researchers knew that the main reason of the eutrophication is the phosphorus loading. So the Shiga Prefecture had decided to cut phosphorus loading to Lake Biwa, first. And then after that, Japanese government watched that, and they, okay, the Japanese government had decided that we cut the phosphorus loading. So the first action was made by Shiga Prefecture, followed by Japanese government. So the Shiga Prefecture was very great to reduce the eutrophication. And then Shiga Prefecture government had been taken many measures to improve the water quality of Lake Biwa. But as you know that only local government effort was not enough to improve the water quality. So especially for the general public effort, that was very important to improve the Lake Biwa water quality. So not only the local government, but also general public and the researcher and the industry people, all those four groups that made a big effort to improve the Lake Biwa water quality improvement. Okay, and due to that effort, phosphorus loading, this is the phosphorus loading from the inlet water to Lake Biwa. You can see here the decrease, even if it is since 1985, decreased so much. And this is also the case for nitrogen decrease, and this is COD. COD is the chemical oxygen demand. Chemical oxygen demand is the oxygen amount which necessary for the degradation of the organic matter within the water. So high COD value means the high organic matter content in lake water. High organic matter content means the dirty water. If I say that very easily. And the low COD value means the clear water, okay? So the inlet water COD value also has been decreased so much. This decrease was due to the efforts by the local government, general public, and the industry and researchers. And due to that, look at this. We have the less occurrence of the red tide since the 1980 to the present. And for cyanobacterial bloom, microcyst bloom, for some reason, we cannot have the clear results, but we have the tendency of decrease of the microcyst bloom like this. But it's very mysterious. The last year, 2016, we had the worst case of the microcyst bloom. So we don't know yet they got the reason for that. But anyhow, we had a good tendency of the decreasing the cyanobacterial bloom here. Oh, thank you. And you can see here, this is the changes in the total phosphorus concentration in the northern basin, big basin of Lake Biwa. As we can see here, that this figure is the white, so it's not so easy for you to detect, but the phosphorus, please take a look at the blue one. The phosphorus concentration in the surface water has been decreasing little by little because Lake Biwa is very big, and retention time means the water coming into the lake, the time needed for the water coming to the lake to go out. If they calculate the calculation, it takes the 5 years for the inlet water come out, 5 years. But for phosphorus, because Lake Biwa is phosphorus limited, phosphorus low amount lake, so every organism needs the phosphorus. So the phosphorus retention time is longer than 20 years. So it takes time to have the reduction of phosphorus concentration. And but anyhow, we started the water quality improvement in 1970s. And after the 10 years, 20 years, 30 years, we could detect the decrease in the total phosphorus concentration like this. And also, if when we success to reduce the phosphorus concentration, the phosphorus is something like the food or fertilizer for the phytoplankton. So we can see here the chlorophyll concentration. Please take a look at the circle. The phosphorus chlorophyll concentration decreasing so much in the southern basin and also in the northern basin. Chlorophyll is the photosynthetic pigment. So this is the data of the phytoplankton biomass in lakes and also in the oceans. So if we have the decrease in the chlorophyll concentration, this means we have the decrease in the biomass of the phytoplankton. So due to the reduction of phosphorus concentration, the phytoplankton lost their food. So they had to decrease their biomass like this. This means we have the improvement of the water quality. So this is why we had the improvement of the transparency like this. Okay? So during the last maybe 20, during the last 20 or 30 years, the water quality of Lake Biwa has been improved so much. But it's very mysterious. We have the increase in the COD like this. Please take a look at the blue one. You can see here the COD concentration has been increased year by year, little by little, like this. As I have already told you that this high COD means the high organic content of lake water. It's maybe the dirty water. And the low COD value means the low content organic matter in lake water. This means the clear water. But this tendency, this result shows us that the water quality or organic matter loading in Lake Biwa are getting worse since the 1980s. It's very mysterious. And the worst case, the Japanese government thinks this is the most important criteria for the Japanese government to judge the water is clean or dirty. This is the most important item for the government. So even if the water quality in Lake Biwa has been improved, that was sought by the sugar producer and the general public, but government, our national government, think that Lake Biwa water quality getting worse year by year. Okay. So, but when we look back for the COD in inlet water, inlet from river water, we can see here the COD loading to Lake Biwa has been decreased so much. This is external loading. External means outside from Lake Biwa. Outside of Lake Biwa, organic matter loading has been decreased so much. But COD in Lake Biwa itself increased. This means this organic matter is not originated from the outside of Lake Biwa, but this organic matter is produced within the Lake Biwa. Within Lake Biwa means the phytoplankton primary production have produced this organic matter. Okay? This is very important. So this due to increase the organic matter from the Lake Biwa. Okay? And the organic matter is most probably produced within Lake Biwa through phytoplankton photosynthesis, primary production. So that is the background of water quality of Lake Biwa. We have the water improvement, but still we have the increase in the COD value. But independently, Kyoto University have been conducting the research in Lake Biwa because our Center for ecological research is originated from the old hydrobiological station established in the 1914. So we, Kyoto University, have the longer than 100 years of the research history in Lake Biwa. And we have been conducting the only basic research, basic research in Lake Biwa. And this is our research vessel, Hasu. This research vessel is equipped with the very sophisticated, the expensive lake or ocean measurement instruments. And at least every week, we come to the Lake Biwa, the long-term monitoring point here, and they collect the water sample and measure the many items. And this is, okay, from now I would like to talk about my research topic. This is my main research topic, Microbial Loop. As you can, maybe you know that there is the food chain, not only terrestrial, but also in the aquatic system, like this. Phytoplankton, it's a primary producer, is grazed by the zooplankton, and the zooplankton is eaten by the fish or some other animals. This is a normal classical or grazing food chain. Okay? But there is another important food chain in the ocean and the lakes. That is a microbial loop. The phytoplankton produce their biomass, but another important organic matter production is the dissolved organic matter. So when they conduct their photosynthesis, they produce their biomass together with the dissolved organic matter. And the dissolved organic matter is released through the photosynthesis. Okay? But this is something like a juice. Yeah, not particulate. This is dissolved. This is something like juice. But the juice, only juice, we cannot live. Animal cannot live only juice. Okay? So animal have to eat something particulate. But only organisms which can use this sort of organic matter, maybe later I will abbreviate this one as a DOM. Only organisms which eat DOM is a bacteria, heterotrophic bacteria. Okay? And the bacteria produce their biomass eating the DOM, and the bacteria, size of the bacteria very small, only 0.5 micrometer. 0.5 micrometer, maybe you cannot imagine. Please, if some of you have a scale, please take a look at 1 millimeter, okay? Please take a 1 millimeter. Please divide 1 millimeter into 1,000, okay, 1,000. And the half of that is the bacterial size in natural environment, not only in the lakes, but also the marine system, okay? Only their size is only 0.5 micrometer. So most of the animals cannot eat, cannot consume those small, tiny organisms. But protozoans, protists, can eat them. So there is another important food chain between bacteria and protozoans. This is called the microbial loop. And after eating the bacteria, protists, protozoans produce their biomass. And their size, protozoan size, would be 5 micrometer or 20, 30 or something like that. So that's why after that, so the protists can be eaten by zooplankton. Okay? So there is, okay, now this microbial something can come to the classical food chain. So this is a microbial loop. And I have been studying about the microbial loop for longer than 20 years. Okay? This is another important thing. Please know this. Thank you very much. And let me show you how the bacteria look like. This is the bacteria in Lake Biwa, stained with the acridine orange, very old type of fluorescent dye. They observed under the epifluorescence microscope. And yeah, as we can see here, the very small dots, yellowish dots. These are heterotrophic bacteria, very small, size of 0.5 to 1 micrometer. The bigger one is a picocyan bacteria. It's an algae, cyanobacteria. Size of the 2 micron or maybe larger. Okay? But anyhow, the small dots are the heterotrophic bacteria. And this is flagellates, protozoans. The flagellates are most important grazer, most important consumer for bacteria. So most of the bacterial production is consumed by the grazing by the nanoflagellates. Okay? Their size is maybe a 5 micrometer or 10 micrometer something, and their concentration, okay, I have to tell about their concentration. A bacterial density or concentration, 1 milliliter is 1 million to 10 million. Okay? Yeah, maybe you can see the water here. Only 1 milliliter water contains 1 million to 10 million cells of bacteria. It's normal. It's normal. Not dangerous. You can drink. Yeah. But please avoid drinking directly. Yeah. And the flagellate density is 100 to 10,000 cells per milliliter. This is very huge amount, but this is normal in lakes and the oceans. Please, next, please. And okay, I would like to show you how the flagellates move. Okay, this is one of the important bacterial grazer flagellates, Bordeaux. Yeah, maybe some of you feel uncomfortable. It's not so cute. It's a bit strange. Yeah, especially after the breakfast. But please allow me to show you this. Okay, this is the flagellates. Okay, okay. Please next slide, please. Okay, the flagellates do a raptorial feeding. They use their flagella to eat bacteria. Maybe some of you think that this flagella is tails. That is not tail. The flagella move like this, okay? This. They take bacteria using their flagella and then eat, okay? Their mouth is located in the base of the flagella, okay? Next slide, please. Okay, let me show you the changes in seasonal changes in the DOM concentration in Lake Biwa. One of my Indian students was interested in the seasonal dynamics of the DOM, dissolved organic matter, in Lake Biwa. He measured the vertical seasonal changes of DOM, and he found that during the summer, we had the accumulation of DOM in the surface water. This means the high photosynthesis due to the phytoplankton produce the DOM and reduce DOM in the lake water. And only DOM is a mixture of the organic matter. So we measured the DOM, the protein one and the humic one. Okay? And we can see here, if we measure the protein-like DOM, this also accumulated in the surface water. But if we measure the humic substance DOM, it's accumulated a bit deeper, together with the very deep water like this, 70-meter depth. And that Indian student, they examined the decomposition of this DOM using some experimental system, and he examined the relationship between the oxygen amount needed for the decomposition of the DOM together with the nitrogen or phosphorus concentration of amount necessary for decomposition of DOM. After the several or many examinations, he had concluded that this DOM, a humic DOM, was produced by bacteria. It's strange, yeah, because the bacteria use DOM as a food and produce their biomass, but they usually not so easily decomposed. So the bacteria cannot eat the humic like DOM. So they eat protein, but they release the humic substances. And then humic substances not so easily degradable, so these substances remaining within the lake water like this. And this is one explanation. This one, deeper one is a bit different process. When the phytoplankton produce their biomass in the surface water, the many phytoplankton cells sink down to the bottom. And when they come to the bottom water, also bacteria, they decompose those phytoplankton biomass. The phytoplankton biomass also contains much amount of protein. So after the bacterial decomposition, the bacteria again release the humic substances like this. During winter time, Lake Biwa water mix. Later I will explain that. Yeah, and they mix. So some of the humic substances accumulated in the surface water would be transported to here during the winter. So that due to those two processes, the Lake Biwa bottom water has the high accumulation of the humic matter accumulation. And please remember the first part of my talk. The Lake Biwa water quality has been improved so much, but we have the problem of the increase of the COD value. So COD is dominated by the DOM. And we, Kyoto University team, think that the increase of COD is maybe the accumulation of those humic-like substances. And this research is a collaboration research between Kyoto University and the Shiga Prefecture. Shiga Prefecture has a very excellent institution, Lake Biwa Environmental Research Center. And there is one good, high-quality geochemistry researcher. So we conducted the collaboration research with him. And finally, we means the Kyoto University and the Shiga Prefecture has concluded that this accumulation of the humic substances would be the one reason of the COD increase. Next, please. And I'd like to talk about another interesting story. Okay, let me introduce you the mixing regime of Lake Biwa. Yes. Lake Biwa is stratified during summer. Stratified means the warm water surface, the cold, heavy water bottom. Okay, but during the winter, due to the low temperature, so Lake Biwa water is mixed well. After that, when it gets warmer, the Lake Biwa stratified again. This is the stratification, mixing, stratification, mixing. This is the annual cycle of mixing regime of Lake Biwa. And this is a figure of the bacterial abundance. And as you can see here, during the stratification period, the high density of bacteria is in the surface water, and it mixed well, and the high density bacteria surface water. This is the cycle. And in some lake, the deep water during the stratification is anoxic, but Lake Biwa is not the case. Lake Biwa, even if the stratification period in the deep water, we have plenty of oxygen here. Okay, next one, please. So one student said this banana-shaped bacteria would consist of the many species. Maybe you can imagine that bacteria is very tiny, many high biodiversity organisms. So that we also expected that the banana bacteria consisted of the many species. So but at that time we were very poor, not so many money we had. So that we constructed only one oligonucleotide probe to detect only one species of bacteria, and they applied it to Lake Biwa water. So finally, we found that all the banana bacteria stained very well like this. So firstly, we expected that the banana bacteria consisted of the many species, but we were wrong. This banana bacteria, only one species. And please take a look at this blue one. Blue one is a normal bacteria, other bacteria. Other bacteria is small, but this banana bacteria stain this one is much bigger than the normal bacteria. And after some DNA analysis, we have found that the name of that bacteria is, please one back. Okay, the name of the bacteria is CN511. Still something like a code name. Those banana bacteria does not have any name at this stage. Okay, please, next. But banana bacteria belongs to the chloroflexi bacteria. But chloroflexi bacteria is usually found in an anoxic environment. But as I told you before, Lake Biwa bottom water is very pretty of the oxygen. So very oxic, not anoxic. So it's very mysterious for us that the CL511 bacteria was found in an oxic environment. Okay, next, please. So my student Okazaki calculated the percentage of the cell banana bacteria compared with the total bacteria. The percentage was 13 or 15 or something. But please remember, 3 minutes, okay. Please remember, the banana bacteria is much larger than the normal bacteria. So if we calculate this percentage in a biomass, this would be 30% or around 50%. So in the bottom water Lake Biwa, it's predominantly predominated by the banana bacteria. And this is not only in our fixed station, but also the Okazaki examined the whole station of the Lake Biwa, and he has found that this occurred in whole northern basin of Lake Biwa during the summertime. Okay, next one, please. And also, another Indian student found that the flagellates, the important grades of bacteria, bloomed in the surface in the March or the June. And the next one, please. But if we apply another oligonoctural probe to the flagellates, we have found that the kinetoplastid flagellates, the video one, that dominated in the same place, the hypolimit, deeper water during the stratified period in Lake Biwa. So next one, please. So we have concluded that there is an important story in Lake Biwa, that during the summertime, the phytoplankton primary production produced the DOM, and the DOM converted into the humic one, and also we have the dissemination of the phytoplankton, and the bacteria. We have been conducting the next generation sequencing of the bacterial diversity in Lake Biwa. As we can see here, some bacteria dominate throughout the year in the shallow to the deep part, but like CL511 only dominates in the deeper water. We have been collecting this type of seasonal changes for each species of bacteria using the HGS. Next one, please. And we have been conducting the biodiversity for protozoans. As we can see here, Illumina, the Lake Biwa protozoans consist of the many species and the phyla or groups. And we also use the codfish oleonucleotide probe technique to follow the seasonal changes in the cell density of the specific protozoan species like this, to examine the microbial loop in the hyperluminum together with the shallow part of Lake Biwa. Next one, please. Sorry, last part of my talk, because time is really limited, I was rushed to finish my talk. Maybe not so easy for you to understand what I'm talking about, but I hope you'd enjoy my talk. Thank you very much. Terima kasih.
Thank you, Prof. Nakano. I think we just heard how Japanese can recover the water quality of Lake Biwa. It's getting worse. The water quality may be getting worse during Japanese developed their industry, I think, at around 60, and then it recovered back after the technology and the knowledge been known till now. So the time now for question time, because of very limited time, we only invite one question for one participant, but after break, I think we still have time if you want to talk to Professor Nakano before he leave for Surabaya. Okay. Your name, please. And
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
My name is Dwi Yitno from the Ministry of Marine Affairs and Fisheries. Thank you very much for the very nice and important lecture, Professor. So my first question is there any potential or sources of fishery in this lake, either wild fish or aquaculture, for example, because we have quite a lot of lake with aquaculture. And then the second question is, the change of the biodiversity in the microbe and then to the protists, did you identify also if this change of the biodiversity affect also to the next step in the biodiversity of microbenthos, macrobenthos, and then in the fish, for example, the wild fish? Do they change also, do they affect also to the biodiversity of the higher food chain? And then concerning the organic matter, did you also identify which kind of organic sources of perhaps there are also organic contaminants that contaminate this lake, beside the very general parameter nitrogen, phosphate, and chlorophyll A, because normally if the industrial sources and then public which they have the different kind of organic type of the contaminant, like LAB and so on.
Terima kasih for your question. If I correctly understand your question, first question would be, yeah, this is a, okay, last one. Okay, this is the changes in the fish crop, fish catch in Lake Biwa from the 1960 to the 2010. As we can see here, our fish production has been decreased so much. Yeah, due to the water quality improvement, that means the cut of the phosphorus loading, that means the reduction of the phytoplankton, but reduction of phytoplankton means reduction of the zooplankton production. If we know the important message is that please do not make the water clean so much. If you clean the water so much, you would have the reduction of the biology in lake. You would have the reduction of the fisheries. Okay, so there is a good. We should have the good balance between the fishery and the water quality. If too much quality, too much clean quality, that means the low biological production, low production of fish. Yes, and another one is if we we can determine the biodiversity of bacteria, protozoans, and phytoplankton, zooplankton, something. So we can know the ecology of each species, specific level, so that we can detect which species of bacteria is more important to the finally fisheries, or which species of protozoan is finally important to the fish, or something like that. So now the sugar prefecture wants to, or the Kyoto Mess also wants to have the good knowledge to understand the, what kind of the food chain or matter cycling would be important for the biological production in Lake Biwa. And the final one would be, yes, this is the data from the Shiga Prefecture. Yes, yeah, this, it's a point source. Okay, sorry. Yeah, the Shiga Prefecture has determined the loading for the non-point source and the point source. And for the point source, it's, for example, the agriculture and the farming and the industry and the house or something like that. So Sugar Prefecture has been collecting this kind of the information. So we can, we know the, which source would be important for the loading of Lake Biwa. Is it correct for your good response to your question? Not English, sorry, very sorry. Yeah, I should have correct, yeah, changed into the English. Very sorry. Sorry, I don't know the Indonesian language. Yeah, very sorry. Yeah. Thank you very much for your comments. Time is right.
Thank you, dear all participants, but because we don't have more time to discuss with very interesting subject with I hope you still can ask a question later on while on the break, because I think you will be here after. Oh, yeah, he will be here until 10:30. So during the break, we can talk to Professor Nakano. Okay, thank you, Prof. Nakano, and give big applause for him.
The second session is plenary session 2. The speaker is Professor Inocencio Ibuot from Botany, Ecology and Biodiversity, University of Philippines, with the same moderator, Mr. Ahmad Saifullah, PhD.
Okay, Bapak Ibu, we will, ladies and gentlemen, we continue with the second presenter for this morning session. We will have Professor Buot from Filipina. He will talk about his background education is in ecology and plant geography. Okay. I don't have your. Professor Buot will talk about. I'm not sure with the title. Ecology. Yes, it's very close related to the first presenter. That's about environment. This time, I think it's direct to give Professor Buot time to present and discussion no more than 45 minutes. Your time now.
To our moderators, thank you so much, and greetings to everybody. And I'd like to thank the organizers for sustaining this international conference on life and biological sciences. This is a very important forum for every one of us to converge. And most especially, I would like to thank the organizer for allocating time, a space for ecology, like the one that was presented by Professor Nakano and my paper right now, to be presented in this forum. This is a recognition of the great contribution of ecology as a science in human society. So thank you so much. I'll be discussing about the terrestrial ecosystem this time. Previously, it was on the aquatic ecosystem. In my paper, I'll be focusing on the terrestrial ecosystem. How can I move? Okay. Yeah. Okay. So, sound ecology, you see, sound ecology actually sustains rich biodiversity, resulting to abundant ecosystem services to human society, which we are all after of. And ecosystem services would include food, of course, medicine, water, air, fresh water, I would like to specify, fresh air, and many others, the cultural values and all. These are ecosystem services. And if you have these ecosystem services, then you will have a very good environmental health. And the environmental health actually is a complex concept. No? It is an interaction of the human health, politico-economic health, sociocultural health, the physical health, and the biological health. Human health be included because we will be the think tank in proposing decisions, appropriate decisions for our ecosystems. And then the political-economic, because they would be the one to execute policies, as what Professor Nakano was discussing earlier. Shiga Prefecture, it's a very sound political-economic health. And all the rest, there has to be a unison. There has to be a harmony of these different components of environmental health. But what is happening right now in the Philippines? There is cutting, there is weed farming, and a lot of things has been altered in the forest ecosystem, which would cascade into the different ecosystems which would depend on the forest, like the river, the lakes, the oceans, so that once there will be cutting and logging in the upper ecosystems, in the upper level, then there will be pollution in the river because nothing would be able to absorb the nutrients already in the upper level. So therefore, it will cascade into the rivers. We will not be able to drink the waters in the springs. And then there will be eutrophication in the lakes and in the oceans, and there will be problems, the red tide that was discussed by Professor Nakano. So that it would mean that all of these five components environmental health should be in accord, should be in unison, should be harmonious in all of these interactions, so that there would be this stability, there would be stability of the environmental health. So that we were investigating different forest ecosystems in the Philippines, and we tried to, in this paper, I would like to try to give a synthesis and then provide some appropriate strategies in order to address the gap that we had found out. So we start our investigation in Mount Pulag. This is actually the highest mountain peak in the Luzon Island, the second highest in the entire Philippines. The first is Mount Apo, found in Mindanao, and this is almost 3,000 meters above sea level. It has a very nice facade, and tourists and scientists are attracted alike into this mountain to study, but it has a very extensive montane forest, the most extensive I've ever seen in the Luzon Island, because most of my trips are in the Luzon Island because we are based in Luzon. I have seen several mountain forests in the Visayas as well, but the most extensive so far is this of Mount Pulag. However, if we take a closer look, there are lots of Sweden farms inside, and there are lots of landslides, because once the Sweden farms' fertility has been depleted, farmers will have to leave, and then they have to cut some more forest lands, and there will be landslides and all. So that when I examined the zonation pattern of the forest landscape, it was very different from what was expected about tropical rainforest. So that in the lower altitude, you will have several pure pine zones, which is not so good. And then in the upper altitude, you will see the oaks, and the oaks are the ones that are being cut by people. So that pines are encroaching into higher altitudes this time. You will find out that pines are encroaching into higher altitudes because people are planting temperate vegetables in the higher altitudes. This is about 2,500 meters above sea level. So they can plant semi-temperate vegetable crops, and then they leave the farm once the soils are depleted, and they cut some more forest, oak forest that is. And then once they leave the farm, the oak will not be able to recolonize. The pines will be able to colonize. That's why the pines are encroaching into higher altitude. And you will see the structural characteristics of the altitudinal vegetation zones of Mount Pulag. It's very anomalous, so that in the lower zones, we have a very low species diversity. The Shannon index, very low in the lower zones. Unexpected of a tropical rainforest. Normally the lower zones should be very high in species diversity and becoming lower towards the upper zones. We tried to examine Mount Akiki, which is almost 3,000 meters above sea level as well. Very steep slopes, but yet you will see agricultural farms in the upper slopes, and therefore, just like Mount Pulag, many of the pines are the ones colonizing once the farmers are leaving the exploited oak forests. So you will see here, the pines are encroaching into higher altitudes. And the structural characteristics are again very anomalous and unexpected of a tropical rainforest. I tried to examine in the southern part of Luzon Island. So this, the species here are more Malaysian in nature. In the Philippines, we have three floristic elements. We have the Asiatic elements in the northern part of the Philippines, and we have the Malaysian elements in the southern part, and some Australasian elements scattered all throughout the island. And so in Mount Mayon, this is more Malaysian in nature, and I tried to see the inside of the forest. There were intensive farming systems inside the forest because people were going up and make their way to make a living in the upper altitudes. And many people are also gathering resources, over-collecting resources, and these resources are very important. We have we have endemic Grammatophyllum species of orchid here, but they are much exploited. When I went into the inside of the forest, only very few branches were colonized by Grammatophyllum already. And added to that, Mount Mayon is a very active volcano erupting every 6 to 10 years regularly. So therefore, it has impacted on the biota as well. So therefore, the mossy forest in Mount Mayon actually is not well developed. I could see because we went into the upper altitude, into the peak, of course, and then I found out that it's more sandy because of the lava, regular lava flow, so it's more sandy on the slopes. And you will see here, there is natural and anthropogenic disturbances occurring in Mount 4. It's quite bald viewed from afar. And so you will have here the anomalous zonation pattern again. The lower dominants are now occupying the upper slopes to replace the upper dominants that were cut by the farmers. Yeah. And then this is the same trend or pattern in Palawan Peninsula, in the Palawan Island, which is known as the last frontier of biodiversity in the Philippines. Yet, this is also occurring in the Aborlan-Guba system. One of my PhD students was working on the altitudinal zones of the Palawan Aborlan-Guba system, more or less like Satuyama. Have you heard of Satuyama? This is a global initiative approved by the Conference of the Parties of the Convention on Biological Diversity in 2010. And you will see here, there is this altered vegetation zonation. The same is true in Mount Tabunan, a remnant primary forest in Cebu City. This is a limestone forest, and it's very important, the home to the endemic Sidamomum cebuense, only found in Cebu City. I tried to highlight Mount Makiling because Mount Makiling is managed by the University of the Philippines, my university. This was started being managed by the university in 1910 when the Americans established the University of the Philippines in 1910. At the foothills of Mount Makiling. Mount Makiling is actually the living laboratory of the natural sciences of the University of the Philippines. This is in Los Baños, Laguna, an hour or so from Manila, south of Manila. This is the so much botanized and scientifically studied because this is the first massive found outside of Manila, so that American and European and Spanish botanists coming to the Philippines in the earlier part of the 15th century, they flocked to Mount Makiling because of the proximity to Manila. But there was a problem. In the 1940s, there were loggings. There was logging done in the 1940s so that our famous dipterocarps, Shoria, Parashoria, and all of these are the dipterocarps. We call this the Philippine mahogany in commerce, but actually in botany, we call these dipterocarps, belonging to family Dipterocarpaceae. And we found out that these were logged during the 1940s, and therefore, this was replaced by Diplodiscus, actually. So Diplodiscus dominating in the lower altitudes, going to the upper altitude, replacing or succeeding, if we want to use the term succession, succeeding the, becoming a seral stage, no? Becoming a seral stage in the absence of the dipterocarp because these were logged over in the 1940s. So the dominants extend distribution range. In contrast, altitudinal zonation publication that was published by Brown in 1919, where there was a perfect zonation pattern expected of a tropical rainforest. As of now, in 2005, when I conducted fieldwork, I found out that the dipterocarps in natural habitat, of course we have lots of dipterocarps right now in Mount Makiling, but they are all planted. They are all planted. But when I went into the natural regenerated dipterocarp seedlings, they were still occupying very low DBA, and therefore it will take time for dipterocarps to recover in the, we do not know what will happen in the future, whether the Diplodiscus will go away or it will stay and dipterocarps will not overcome diplodocus, just like the pinus of Mount Pulag. So that in that study which I conducted on the different land use types in Mount Makiling, we found out that altitude, among others, distance from disturbances and slopes were the ones influencing the distribution of these land use types. And in another study of one of my PhD students, she found out that elevation, of course, elevation should always be one. Air temperature, which will go with elevation, of course. And then you have the pH, the moisture content, the water holding capacity, the human disturbance, among others, are the ones controlling the distribution, the altitudinal zonation of vegetation. So that if I have to summarize, the data therefore showed that when there will be clearing or disturbance in the upper elevation, of course a gap is being formed, the farm is being established. But when farmers will abandon their farms because of the depletion of the nutrients, upper elevation species are given a chance to recolonize. However, in the case of Mount Pulag and in the northern mountains of Luzon Island, they were not able to reestablish. So that pine forests are the ones dominating right now, just like the Pinus taiwanensis in Taiwan, where they are the ones becoming topo-edaphic climax because of the very steep slopes, just like in the Cordillera mountain range of Mount Pulag. It's a literal Cordillera, actually. So therefore, the oak could never be able to regain dominance in the former oak forest per analysis of our study. That's why the pines are now existing there for several years, several decades already. The pines have been there. And then this is due, of course, to the change in the microclimate, because once the species are cut, of course, the microclimate had been disturbed. So that there is change. It could be slight or drastic. We were not able to see actual measurements. We base our measurements on the record of the Weather Bureau of the Philippines using several computations. And then we have, they were given opportunity in Mount Pulag because it has been there for a long time, but then the pines are still dominating. I think we'll never give way to the oaks anymore, just like the case of Taiwan. The Pinus taiwanensis of Taiwan, they are dominating now. They are topo-edaphic climax. And then more adapted lower elevation species will have to colonize, of course, because the law of succession, they will really colonize. And then in the case of Mount Makiling, there was the diplodocus colonizing the pterocarp in Mount Makiling. So that we can have I would like to discuss our research directions. There has to be detailed studies on succession in heavily disturbed habitat. Just like in the case of Pinus, more or less, because it has been there since 1900s, there were several pine forests already because people had clearing the forests. And then we found out that Pinus are now establishing dominance in the former oak forests. But in Mount Makiling, we do not know what is happening because the original dominants are still occupying. They are regenerating, but they are occupying lower DBH classes yet until now. So therefore, we would like to study the succession in heavily or forests undergoing heavy disturbances, like in this case, logging. There was severe logging of the pterocarps in the 1940s. And I think there has to be some sort of biodiversity corridor establishment because our forests now are fragmented. And because these fragments have to be connected and reconnected so that the biota, the living organisms will be able to travel with convenience. You will be facilitating regeneration through the establishment of biodiversity corridor. In our work in Ranong, Thailand, one of my PhD students was, because we were really concerned about the tsunami in 2004 in Ranong and the rest of the southern Thailand. And when we went into Ranong coastal ecosystem, we found out that there were no more mangrove forests that could have blocked the strong waves and mellowed the waves before it will struck communities. However, there were none anymore. So that I was asking my PhD student to build corridors because there were some patches and fragments of mangrove forest. And then ecological valuation studies. This is very important so that people would be able to understand why don't we cut recklessly. Why is it so? Here, if we are going to value this economically, we value only the flower, the aesthetic value of the flower. However, if we are going to value this ecologically, we have to identify the pollinators, the nectar, the dependence of the nectar and the dependence of the pollen. They should be included in the computation of the final price of this flower. But then we just buy the flower as it is. We are used to that because we are used to economic valuation. I think ecological valuation should be instilled, no? And in our classes, in ecology classes, in botany classes. Dr. Haris Listiwati of Jember University has been working on this, ecological valuation studies. Just like this. We cut the timber for timber, of course. We cut the tree for timber only. But how about the photosynthetic capacity of the leaves of this tree? How about the capacity of this tree to store the biomass? Photosynthetic products. We did not compute that. We never computed that. We only compute the timber, the board feet, not board foot. But we never computed the ecological value, the organic matter that will be produced by this tree for a number of years. Have we computed that and incorporated into the total price when we cut the tree? And then there has to be massive community biodiversity education among adults, youths, and the kids at all levels, including our political leaders, of course. We are always complaining that our leaders do not know because their fields of specialties are different. So it is our duty as scientists to inform them as well through massive biodiversity education. This time we are in the digital age. We can have education on the internet. So therefore, we can inform our leaders. We can be extra diplomatic, informing our leaders that it has to be so that they will understand. How can we ask them to protect? How can we ask them to protect when they have not even loved our nature and our forests? In fact, in the International Botanical Congress in China, I recently attended, one of the keynote speeches was emphasizing on this, that how can we encourage people to protect our nature when they have not even loved nature? We have to teach them first how to love nature before we can ask them to protect nature. Okay. Also, there has to be more local, regional, and international collaboration, which we are doing right now, actually. Interdisciplinary and transdisciplinary discussions. We cannot just concentrate on biology because, you see, most of our problems are interdisciplinary in nature. So we have to involve one sociologist. We have to involve some artists. It has to be transdisciplinary. We have to involve some engineers. We have to involve some medical doctors and all these disciplines that we have in the university so that it will be more holistic in approach. And then there has to be intergenerational cooperation because old people will retire and we die. So therefore, we have to have intergenerational cooperation at the Institute of Biological Sciences, where I belong, in the University of the Philippines. We have our understudies. We are required. Senior faculty members are required always to have understudy. So I have one understudy, an assistant professor, who would always be undertaking. We always collaborate in research and instruction. So we teach the same course. We co-teach courses in one semester, two of us teaching one course, and then doing research together as well, so that there is already intergenerational cooperation occurring in the university, in the institute level actually, so that we will have stable environmental health. Once all of this would be in accord, in unison, in harmony, I think there will be stable environmental health and lots of ecosystem services for human society. I have my collaborators here, and thank you so much.
Thank you, Prof. Buat. We still have about 10 minutes discussion, more than 10 minutes discussion. So we invite at the moment 3 questions from 3 participants. Okay, I give for the first question for participant in the front, and the second over there, and the third question from. Okay, at the moment we give to participant to ask a question. You can give direct answer.
Maraming salamat. Magandang umaga. My name is Arin Afril. I'm from Sriwijaya University, Palembang, Indonesia. 4 years ago, I visited CLSU, Central Luzon, and then we learned about the biodiversity in this Luzon island. I would like to know about the progress about the invasive alien species as a threat for forest ecosystem in the Philippines. Do you have any ideas about that? Thank you. Invasive alien species.
Okay. Actually, there are some groups who are working on invasive species in the Philippines and in Mount Makiling particularly, because because you see many of the nations are using South American reforestation species and many other exotic species brought to our countries. So we have actually, and there are some professors who are documenting on the proliferation of these, like the Swietenia of the Meliaceae family, the Berbinaceae family, the Gymellina. So there are some. We have Alnus japonica. There are lots of these also, and but I think more documented studies are of Gymellina and Swietenia, and they are encroaching into the natural forests. That's why ecologists in the forest of in the College of Forestry and Natural Resources of the University of the Philippines, they are warning the entire university in their publications that we have to keep watch on the proliferation of Gymellina and Swietenia in the natural forest of Mount Makiling.
I hope that's give a clear answer for you. Okay, for the next question, please.
Okay, thank you very much for the opportunity. Sometimes in some tribes or local population, there is also what we call local wisdom that also can be used to conserve the natural resources. What do you think in this case in your country, for example? It is also useful to explore this aspect to help us in conserving our natural resources. What happened in Indonesia in some local population also happened this problem. Thank you. My name is Jamsari from Andalas University, West Sumatra, Indonesia.
Yeah, that is a very good initiative actually in the Philippines, because many people are climbing on mountains during holidays. Holidays, they go up to the mountain and then they meditate. So they are asked to bring their waste back. And some of them are doing reforestation activity as well. some of the groups, but I think these are not mandatory. They are not mandatory, but they are encouraged to do so. There are many groups doing that. Actually, I learned this in Japan because in Hokkaido before, we went to Hokkaido and we bought our laboratory, because I went to the Nature Conservation Laboratory of the University of Chiba. So we always go to different forest ecosystem of Japan. One summer we went to Hokkaido and we bought a lot in Shirotoko. I don't know if Professor Nakano is familiar of this initiative in Shirotoko, that they are selling lots, quote unquote. They sell lots to visitors, and we, and this, our laboratory was able to buy a lot for reforestation. So that, because we will not be able to reforest because we stay there only for 1 week. So that I think that money will be used for reforestation to be spent by the park management. We are not, I have not yet heard of this initiative in the Philippines. All of these are more of organizational initiatives where they just do reforestation on their own, but the park management is not collecting money to, for this purpose, unlike the one I've known in Shirotoko Park in Hokkaido, which is, I think, a very good initiative, because there are lots of, because we cannot just do reforestation in our visit. We go there to enjoy ourselves and to enjoy the forest, and yet we do reforestation. Of course, we can also enjoy doing reforestation activities, but because, especially if we are students, we go there, we study. So therefore, I think that initiative in Shirotoko is very good, where we just pay, and maybe the park management would be able to, will be the one administering in the reforestation, just spending the money we pay.
Yeah. Thank you. It's clear? Okay, we still have, I think, 2 more questions for 2 participants, if possible. Okay. Yep. Because you hold the microphone already, you can ask first, and then the second is Ibu from the right wing.
Okay, thank you. My name is Abdul Ghafur from AAA R&D Indonesia. I know some species of plants and fungi in Australia, formerly or previously only found in the northern part of Australia, now are colonizing the southern part, even further to Tasmania. And people attribute this problem partly to the global warming, to the climate change. Would you comment on this, especially related to your study in the Philippines? Thank you.
Fungi? Yeah. I think so. It could influence, because although fungi are ubiquitous, they are ubiquitous in nature, but maybe there are some species which require certain temperature, or that could thrive, because we have some thermophilic and loves more warm temperature, something like that. So I think so. Of course, I have not heard of that, because I'm studying only the angiosperms, but I think so. It is not impossible. Yeah.
Okay, thank you, Professor. I am Erika Pardede from Nomensen University from Medan. I'd like to ask you about how bad it is actually about deforestation in Philippines. I'm specifically linked to the pine, Pinus mercusi, because there is a similar species from with the one we have in Sumatra. How bad it is, because in Indonesia, especially in my region, the Pinus mercusi extracted illegally, legal also, and it is almost extinct actually. Thank you. How is in your country?
What was the first question?
What's the condition actually in your...
Yes. You were asking about the deforestation of the country? Quite bad. Maybe I would say very bad. Very bad. Because people had been doing Sweden farming for a long time. And maybe during the start of the century, it was just sustainable because there were few people. But now we are 120 strong. So therefore, I think it is not sustainable anymore. It is affecting badly the forest ecosystem. So that's why the government is having the national greening program. The previous administration is doing was initiating the national greening program of the government and throughout the Philippines because of the bad condition of deforestation. Right now, logging concessionaires are, I think we have a temporary stop, but especially with the previous Secretary of the Department of Environment. But you know politics, how politics works, I really do not know what will happen next. Because you see, no matter how ecologists will have to tell them, they still need biodiversity education, as I have emphasized earlier. Yeah. Sadly, it is not that good anymore. The next question is about Pinus mercusi. The Pinus mercusi in the Philippines is distributed in the southern part. So we have in Mindoro, actually, we have in Mindoro Island, which is near to, if we go back to our geological history, there was a connection between Mindoro and the Borneo, something like that. And then Palawan also recently reported that they have Pinus mercusi. Not all of the scientists in the Philippines know that there is Pinus mercusi in Palawan Island as well. It was recently published that there is Pinus mercusi, to the pleasure of botanists in the Siswambana Botanical Garden, because they are now doing a biogeographic study of Pinus mercusi. Of course, this is listed in the Red Book of the Philippines, the Pinus mercusi, and they are now suspecting that the Chinese Pinus is related to this of the Philippines and that of Indonesia. I really do not know what will happen to this collaborative activity that we have with the Siswambana Botanical Garden in southern China. So for the meantime, the distribution range of our Pinus mercusi is in Palawan, Mindoro, and of course, there are some reports that is found also in the central Luzon. The northernmost distribution is Zambales. This is the Caraballo Mountain Range. Caraballo Mountain Range. It's found in Halcon in Mindoro. It's also found in Palawan and in the Caraballo Mountain Range in central Luzon, where Pinus Kisia is also found. So both of these Pinus are found in the Caraballo Mountain Range. But Pinus Kisia does not go southward. The southernmost limit of Pinus Kisia is Caraballo Mountain Range. And the Pinus mercusi's northernmost limit is the Caraballo Mountain Range as well. They converge in Caraballo Mountain Range. Distribution, but not much studies on this biogeography yet. Thank you.
Thank you very much, Prof. Buol. I think we don't have enough time to discuss about the very interesting subject. To sum up this lecture, both by Prof. Nakano and Prof. Buol, I think we are as human need to involve to preserve our environment. There is no reason for us not to getting involved to get participate to preserve our environment. In water, for example, Prof. Nakano show us how regulation and technology can improve water quality, and also from Prof. Bual with programs such as mentioned in Philippines can also improve the vegetation program. That's for me, and then because the time is up, I return back to the committee. Thank you very much for all contribution, your attention, and your dedication. Thank you.
That was the end of the plenary session 2. Ladies and gentlemen, can we give